Welcome everyone, and thank you for joining us for today's webinar, Challenges of Cannabis Testing in the U.S. and Abroad, presented by Deborah Miran, a former commissioner for Maryland Medical Cannabis Commission. My name is Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, simply click on that Ask a Question box and let us know you're experiencing the problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of your presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming our presenter, Deborah Miran. I will now turn the presentation over to her. Welcome, Debbie.
Thank you, Susie, for that kind introduction, and good morning, everyone. I'm delighted to spend the next half hour or so sharing some of my thoughts with you about the challenges of cannabis testing. As Susie mentioned, I have no conflicts of interest. I represent myself as a regulatory consultant to the medical cannabis industry, and I am a former commissioner with the Maryland Medical Cannabis Program. By way of abstract, I'd like to introduce my talk by saying the vast majority of United States and other countries require quality control testing of cannabis and cannabis-produced products prior to release for distribution. The purpose of product testing is to provide patients with the assurance that the medicine is potent and pure and safe for use. Analytical testing also assures that the contents match the label claim, that there is batch-to-batch consistency, and the product is potent and pure throughout shelf life. Each state and country has its own set of testing requirements and regulations for analytical laboratories. With these requirements come challenges for both the regulators as well as the regulated industry. So, by way of learning objectives today, I would expect that those that are listening will learn how medical cannabis products are tested and the quality standards. They will also learn about the regulatory testing requirements in the U.S. and other countries. And finally, we'll appreciate the various testing challenges as states' programs grow and evolve. So, to get into the meat of the talk, I'd like to first begin by saying, what is the purpose of testing? To some, it's self-evident, but the four most important points are the fact that we want to assure, of course, that the products are potent and pure. We want to make sure that the products match the information on the label claim. This is the message to the patients and to the distributors and dispensaries about what is the contents of this product. We want to assure consistency from batch to batch. Many of these products are used chronically. Patients need to have the absolute assurance that from batch to batch, lot to lot, container to container, they're getting the same product. And of course, the ever important potency and purity throughout shelf life. So what are some of these general testing requirements and quality control? Essentially, 27 out of the 33 states that have medical programs require QC testing prior to product release to the dispensary. These laboratories that perform the testing must be independent of the grower, processor, dispensary, or by any licensed authority to participate in a state program. Many states require that the independent laboratory be accredited by a third-party accrediting body. Quality control begins with standardization. And what this means is that you don't test quality into a product. It begins with the step one, which in this case is the planting of the seed or the clone. And this standardization proceeds all the way through to harvest, to packaging, processing, and eventually to the dispensary. I can't emphasize enough that the requirements for standardization be something that is adhered to through the entire process. As far as testing for standards, we apply the good manufacturing practices, good agriculture practices, and of course HACCP when food production is involved. And last but not least, within the laboratory itself, quality assurance programs and up-to-date and complete and form SOPs will ensure consistency within the lab, between operators, and day-to-day consistency. So a few definitions that will be pertinent to the rest of the talk. Obviously, there's many, many more definitions, but these two are particularly important to me. What is an approved laboratory? Well, the approved laboratory is one that has a facility that has applied for and received approval, whether it be an approval or just a registration, a license, whatever the state requirements demand. And this approval is received to identify, collect, handle, and conduct tests on samples from both growers or cultivators and processors. And the second key definition is, what is the scope of accreditation? This is a term that's used by the accrediting body to detail the approved list of test methods with full method validation that the lab has been accredited to perform based on whatever state requirements are. So in other words, the accrediting body does not actually write the methods, nor do they judge the method for fitness. They assess the method and they make sure that this is the method that's going to be used every single time that a sample is tested at this accredited lab. So what are some general provisions that states require when a laboratory wants to start business? First of all, as I previously mentioned, the lab must be approved or registered by the state. Here in Maryland, we register labs. It's an application process. Some states actually have a formal approval process. The regulatory authorities, such as the State Commission, will post on their website a list of all approved labs so that information is available to the general public and to the licensees. Laboratory personnel, including the director, should be trained and educated. Most states have very specific educational and training requirements at the director level, as well as managers and lab agents. Labs are subject to the inspection by state authorities, and so that the state retains the right to come in at any time, whether it's a regular routine inspection or a for-cause inspection, something that would allow the states to come in and do an inspection. Many states require proficiency testing. In other words, blinded samples are prepared and made available to the labs for them to conduct tests to compare against a known quantity to give the further assurance to the state that the labs know what they're doing. With regulations at the state level come penalties for violating procedures. These can range from fines to injunctions and loss of license. We hate to think about falsifying reports, but we know that this can happen, and if it does, it is certainly something that is actionable by a state authority. And finally, some states will collect field samples from time to time to also check on label content and also to check for purity. So after the laboratory has decided that they want to go into a business in a state with a program, here are some of the basic procedural approval steps that most states have. Obviously, these will vary from state to state, but in general, the submitted application will, of course, include the name and the address of the owner, the name of the director of the laboratory, a copy of the certificate of accreditation, or in case the accreditation is still in process, some states, for example, like Maryland, have a provisional period in which the lab is allowed to get up to speed and complete the accreditation. So in those cases, it would be a contract with an accrediting body that would suffice. A complete list of the SOPs that the lab will prepare and use. And I highlight sampling protocol because we're going to spend some time talking about that later on. Very important. A list of all the laboratory equipment, a complete description of all the tests that are going to be performed, going back to that definition of scope of accreditation, a full and complete description of the quality assurance program, chain of custody and sample tracking in states that have track and trace systems. This reaches out to the transfer of samples to and from laboratories from other license holders like the cultivator or the processor. And of course, the competency of the employees, their knowledge and experience, and of course in some states a background check is required as well. So testing requirements, as you can imagine, vary from state to state. And this is just a generalized scheme of what are most commonly seen as the testing requirements in the states that I have done work in. Some states, for example, require a written contract between the licensee, the grower, the processor, and the testing lab. Some states require that the grower, processor, laboratory results are entered into the electronic tracking system, which is controlled by the state program. The scope of testing for each plant harvest and each processed product lot might vary some from state to state, but in general, this is QC. So QC means that every lot or every batch of product that sits on a dispensary shelf has been tested and released and meets these specifications. So at minimum, we have a list here which are, of course, some of the purity requirements, pesticide residue, if in fact pesticides have been used in the cultivation of the plant, solvent residues, if that is part of the extraction process of the cannabinoids and terpenes from the plant material, water activity and moisture content. These are vital to make sure that the plant or the dried flower, if that's the finished product, has adequate shelf life, and that there isn't enough water that would support microbial growth. Microbial contaminants such as pathological bacteria and fungus, aflatoxins. And then on the potency side, of course, the two main cannabinoids, THC and CBD. And most other states expand that list to include some of the minor cannabinoids such as the acid forms of THC and CBD, and also CBN and CBC and some of the others. That would have to be something that would be checked state by state in their various rules. And then terpenes, there's a lot of variability about testing requirements for terpenes. For example, in Maryland, we have a list of 8 that we require the labs to test for, if in fact they are on the label. And that's an important component. If the labeling does not include a description of terpene by content, or if the levels are below the level of detection, then the requirement for testing terpenes is waived. And some states, and this is a very big one and important one for me, is that some states require stability testing. I think this is an area that we're going to see grow. Maryland does require it. Now, as of this year, they have not been enforcing that requirement, but it is my understanding that they will begin to enforce it. And the significance of stability testing is that a base of data will be built. It will be important not only for the grower or processor, but also for the state agencies and actually for the world at large, because there is quite a dearth of actual shelf life data to understand how long these cannabinoids and terpenes remain stable in either the plant product or the finished product if it's processed. So shifting gears now to the second half of the talk, I'd like to talk about some of the challenges. As you can understand, with every program, there's the ideal, which I've just gone through on the first 10 slides, but then there's also reality. Things are going to happen both on the side of the regulators and the side of the regulated industry. And so what I have in the next few slides is 4 examples of what I have witnessed and observed as current challenges in this space. We're going to spend a little time talking about sampling, a very important topic and very timely. The next challenge is something I call granulation versus beautiful bud, and I will explain that. Inflating THC test results. Sadly, this is an issue. And then finally, misrepresenting total yeast and mold test results by virtue of using different analytical techniques for measurement. So let's move to sampling. A sound sampling plan is absolutely vital to make sure and to assure the best possible accuracy in the result, and that result reflects and represents the whole batch. So in other words, this is a plant. This is a plant that's harvested. There'll be plant parts. It'll be visible. There'll be trim. There'll be leaves. There'll be sugar leaves. There'll be fan leaves. And, of course, there'll be the dried flower bud. And all of this is very non-homogeneous. And so collecting a sample of this sort is difficult, and it's difficult to represent it statistically sound. So what most states have done, and I believe Maryland was the first to require this, is that the sample not be collected by the licensee, but rather a representative from the testing lab who is independent and unbiased will come in and perform the sampling, Sometimes the sampling is performed under video surveillance, or at minimum it is performed as a witness with another person witnessing the sampling. As I mentioned, the sample must be representative of the whole batch or lot. So there's various ways that a statistically sound sample can be taken. And if you've got a bin and you've got plant material in it, you can use a sieve, a sampling device, to go down into the middle of the bin, the sides, the top, the bottom of the bin, and collect an aggregate sample that is as representative as it possibly can be. And in states that require stability testing and quality assurance samples, it is my advice that those samples all be collected at the same time the QC sample is taken. So the QA sample is put in retain for a later use if, in fact, there's a recall or there's a patient adverse event. That QA sample can be retrieved and used to retest to reconfirm the product still meets specification. And, of course, the stability sample will be stored in a controlled room temperature environment until the interval at which the testing is required occurs. So, challenge number 2, the granulation versus the beautiful bud. This somewhat overlaps with the sampling dilemma. As I said, when you've got a bin of harvested dried flower, clearly you can see flower buds, you can see leaves, and one of the issues, one of the remedies for this is to actually size the batch. And what do I mean by sizing the batch? The plant material can be put through a grinder or a sizer with Teflon coated or otherwise inert chopping blades so that the sticky material in the trichomes isn't lost to the blade. And the result then is a much more uniform harvest batch. And so sampling then becomes a much easier challenge, and it really does reduce drastically the chance for sampling bias. The dilemma, though, comes in the fact that in the U.S., and this is an observation of mine, and I believe of many others, there is a general patient consumer preference to see an intact flower bud. Quite honestly, as an analytical chemist, I don't understand that, but I guess this is something that has become tradition. And so it is largely unacceptable in the U.S. to have samples in the dispensary that represent granulated flower material if that is the finished product that the patient is interested in. And once again, sampling of that non-granulated flower bud may not be representative of the whole batch and has much more of a chance to have a sampling bias. So, challenge number 3, and this one is a difficult one even to discuss, but I think it's important enough that we spend a few minutes talking about the how and the why. First of all, the how. How can you inflate THC results? You know, the lab has procedures. We've got state rules in place for the most part. Everybody is accredited. It should be a fail-safe system. But here are 5, and I'm not saying these happen, these are potentials, but here are 5 potential ways that I have conceived of can be ways in which you can subtly influence THC results toward a higher level. First of all, something that I call secondary sampling. So the sampling has been done at the licensee. The sample comes back to the laboratory. Absent video surveillance, the lab analyst is there preparing the sample that is going to be ready for the instrument. And so obviously that entire sample that's been collected is not needed. And so secondary sampling, if the product is not granulated, can be potentially biased at this point. Secondly, a grower can pre-select what they are going to make available to the independent sampler before they even arrive. I can't say once again that I have witnessed this or that I know that this practice has taken place, but once again, it is a potential. Thirdly, if the analytical methods lack sufficient validation, you could introduce a bias by virtue of a bad method. Fourthly, something that I call intentional and subtle dilution errors. What does this mean? Well, obviously, instruments are very, very sensitive, and so the sample from preparation to dilution, in order to have the proper concentration for detection and quantitation, will be diluted possibly several times before it's actually introduced into the equipment. During that process, subtle dilution errors, which could add up, could potentially concentrate the product that is going to be injected in the machine, and thus, an unintentional or intentional inflation of the THC results could occur. And last, and probably most egregious, and I hope I would never experience this, but if data after the analysis is completed is exported as a text file and is unprotected from tampering, then results through human intervention could be changed. So the second question then is, why does this occur? Why would there be reason to ever want to inflate THC results? Well, the two most predictable answers that I can think of are things that I've seen in Maryland, once again, and probably in other states as well. And that is, for the most part, pricing of the product, if it's not controlled by state rules, typically is based on THC content. So the grower sells to the processor. That price from the grower to the processor will be higher based on the higher level of THC. Likewise, from the processor to the dispensary, and then ultimately from the dispensary to the patient. And so there is incentive throughout the entire chain to want to present the product with the maximum amount of THC possible. Secondly, and I know this is happening in states, especially with states that have rules about approved conditions that might allow for some more flexibility in physician recommendation. We have seen, and I have to admit, I don't like it, but that there is a recreational component to medical markets. And so if you believe that and if you've witnessed that, then it's understandable that in the recreational space, the higher THC content is obviously what is more desired. So the last and most interesting, I think, challenge that we've seen goes back to an incident that occurred about a year or so ago, and it regarded the test for total yeast and mold. Total yeast and mold is a typical test that is performed on botanicals, plant material, medicinal plant material, and the test typically is it's based on the test that is published in the American Herbal Pharmacopoeia monograph, and that monograph is based on the standardized methods from the Bacteriological Analytical Manual. And typically what is recognized even to this day is what is known as, traditional plate counting on agar plates. So that is the recognized standard at this point in time. But with the advent of PCR testing, PCR standing for polymerase chain reaction testing, it's a very quantitative method. It's very precise. It's very accurate. But it does have some reservations at this point in time, in my opinion. The test is valid for species-specific organisms such as Salmonella and E. coli. So what does that mean? In a total yeast and mold test, basically what you're measuring is any fungal species. So we know that there's thousands and thousands of those. Unless there is a primer, which is the equivalent of a standard for these species, the PCR test, in my opinion, at this point in time, is not completely valid. Thirdly, the PCR has the potential to measure both live and dead DNA of the organism, and that can influence the accuracy of the result. And at this point in time, the BAM, the Biological Analytical Manual, does not consider this a valid test for either total yeast and mold or total aerobic count. So an interesting study was conducted in Nevada. This goes back, I believe, to 2017. And they were having issues, and these issues were coming to the attention of the state by various labs. So the state decided to conduct a very interesting study. And in this study, the state collected 41 samples of cannabis flower. These samples were subjected to testing by both the traditional plating method, as I previously described, as well as the PCR technique. The specification, which is generally recognized and is included in the AHP monograph, is a specification of less than 10,000 colony-forming units. So the samples were prepared for plating based on the BAM method, and for the PCR technique, the samples were prepared based on manufacturer's directions. So the results that came out of this were very interesting. 16 of the 41 samples passed by both techniques. But more importantly, 20 of the 41, so basically half, failed by plating, but then most surprisingly, 3 of the 41, only 3 of the 41, failed by the PCR test. So what did the state conclude from this study? Basically that the PCR technique generated on the average of a result of an 85% false positive rate, and that the PCR test therefore was not considered valid. And as a consequence, the state of Nevada has now banned the PCR as a technique for measuring either total yeast and mold or total aerobic count until such time as the method can be fully validated by a recognized authority such as the AOAC or ASTM or some other testing organization that performs this kind of work. And another conclusion that came out of this test from Nevada was that states that came on board downstream, such as California, took a look at this and said, well, you know, what's really important? It is what is pathogenic to humans. And so California opted to drop the test altogether in favor of species-specific pathogenic tests with limits, such as we mentioned, like Salmonella and E. coli. So that really was a very important study, and I believe that as other states come on board, they will be taking a look at this. And at some point in time, I would hope to see the PCR test become the recognized standard as we advance science. So now for the remaining time and the remaining slides that I have, I'm going to shift gears and talk very briefly about some key international markets. These are places where we know they have national laws, and I'm referring mostly now to just medical use. And so let me start with requirements in Canada. As we all know, Canada has had a sea change with the update in last fall with the inclusion of adult use as a national law. But they also revised many of their medical regulations. And so in 2018, the regulations for all testing, both adult use and for medical use, were revised to require that each batch or each lot of product must be tested, plain and simple. The requirements include, but are not limited to, potency, meaning THC and CBD content, and purity testing for things like heavy metals, pesticide residue, and microbiological contaminants. The Health Canada recognizes 21 permitted pesticides that are allowed to be used, and the Health Canada authorities will conduct random testing from time to time on products to make sure that these pesticides, if they are present, if they are being used, do not exceed the tolerance limits that Health Canada has established. And finally, while Health Canada does not accredit testing labs, they do license them. And at the time that I published this presentation, I believe there were upward of 52 licensed laboratories in the country. Moving on to the Netherlands, a country that I particularly admire, and I have done a lot of studying there. The Netherlands have had medical use as a national law for a long time. They have absolutely perfected the process of growing, processing in a standardized way. And likewise, they have a very, very thorough and strict, efficient testing program. So under the Office of Medicinal Cannabis in the Netherlands, there is a requirement that each lot and each batch of product, once again, be tested. And the testing is done by an independent lab contracted with by the Dutch government, and I believe that company is known as Pharmalease. And requirements in the Netherlands, once again, are what you would consider standard: potency, water content, loss on drying, heavy metals, pesticide residue absent, microbiological contaminants, and also appearance. The one thing I will add too about requirements in the Netherlands is that potency has very tight specifications around the label claim. So in other words, if the dried flower is the finished product and the dried flower is purported to have 10% THC, the specifications in the Netherlands are plus or minus 10% label claims. So a range around that 10 would be, of course, 9 to 11%. And those strict criteria are what would be equivalent to what you would see with an approved drug product. And so you can see they're very specific, very strict. And part of the reason for this is the years and years of experience of growing in a standardized way and perfecting analytical techniques that they are able to impose these kind of strict specifications. And I would say that this is a direction that all testing programs should be headed towards someday. Moving on to my final country, which is Australia. Obviously, there are many, many more countries that have national laws and testing requirements, but I wanted to just highlight Australia because they have a program that's finally off the ground and growing very rapidly. And under the Therapeutic Goods Administration, Therapeutic Good Order 93 requires, once again, that each batch, each lot of product is tested. And this includes product that is exported to other countries and also product that is imported from other countries. Nothing sits on a dispensary shelf or a pharmacy shelf in Australia that hasn't been tested and released. So what does Australia rely on as far as standards? Basically what they have done is to adopt portions of the European pharmacopoeia, just like we have in the U.S. with the AHP monograph, for standardized procedures for things like residual solvents and heavy metals, and also preparation and for extracts and dried product based on medicinal herbal products. Required in Australia are, of course, potency testing. And here you can see I have in parentheses the specifications. Slightly different from what I've just described in the Netherlands. For flour, the range is a little more liberal. So in other words, if you're between 80 and 120% of that label claim, so going back to the example of a 10 milligram, or a 10% THC content, in Australia, potency would pass if it ranged between 8 and 12%. However, for dosage forms, which obviously have been processed from the flower and extracted and formulated into traditional dosage forms, Australia then reverts back to the traditional 90 to 110% label claim. So I think this is a very fair approach, and I would expect that this matures and standardization of the whole process ensues. that once again, too, Australia will tighten up the range around potency for dried flower. Australia also requires that heavy metal testing and pesticide residue, microbial contamination, and also an appearance test, which would include foreign matter and a test for total ash. So I'm going to wrap up my talk now with what is next, what is in the future. This industry is rapidly evolving. I don't even think it's evolving, I think it's revolving at the speed at which everything is changing, not just in the marketplace, in the regulatory environment, but also in the testing space. We know there are many organizations right now, including the United States Pharmacopoeia, the European Pharmacopoeia, and other private organizations, such as I previously mentioned, AOAC, the American Chemical Society, ASTM, are all currently very, very involved in working on drafting a national and an international standard for some of the tests that we've talked about today. I look forward to this. I think this is a largely positive development to have one standard. It will help the industry, it will help the testing labs, it will help regulators, it will help everybody to have one standard. And a standard isn't static. A standard is something that is basically as good as it is until the science and the technology grows. And we know that will always continue. And so the standards are meant to be reviewed from time to time on a periodic basis to look at currency versus, the state of the art and the state of the science. I think we're going to see much more enforcement and requirements for compliance at the state levels. Many of these state programs like Maryland are just getting through their initial year or two of operation. And so the states now have a chance to sort of refine and get more involved in enforcement and inspection requirements so that some of the challenges that I've mentioned earlier maybe won't be challenges at some point in time. And there will be a good symbiosis between the regulated and the regulatory. And finally, I think that we've seen it already, some of the accrediting bodies, some of these independent, for-profit or not-for-profit accreditation bodies are beginning to partner with groups, and including state regulatory bodies, to make the accreditation process more cannabis specific. And so that it will just make the accreditation even more meaningful than it is right now. So with that, I thank you very much for your kind attention, and I will be delighted to respond to any questions you have by email on my talk, my presentations, and any aspect of testing regulations. This is something that I am very passionate about, and I hope that you have benefited from this talk today and have learned something at least enough to forward some important questions to me that I will be happy to answer. And so with that, I will sign off and return this portion of the talk back to Susie. Thank you again.
And thank you, Debbie, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that this webcast will be available for on-demand viewing through June 2019. As a final reminder, Debbie will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We'll see you next time. Bye-bye.